how's it going everyone? It's Chow here, and today we are going to discuss a little bit about restriction enzymes, so let's get started. Okay, so this is going to be super brief, but restriction enzymes are basically these enzymes that are found within many organisms that are basically used to cut stuff. That's basically the easiest definition I can give. Now, restriction enzymes in bacteria, for instance, are actually going to be used sometimes to prevent viral infection by degrading certain sequences like DNA from the viruses. So we're basically taking this enzyme, this protein, to go in and recognize a specific site on the DNA called a restriction site, and they're going to do some cutting. There are kind of two main groups of restriction enzymes, one group being called the endonucleases and the other being the exonucleases. The main difference between endonuclease and exonuclease is the location where they cut fragments within the cell. So endonucleases are going to cut fragments within the cell, so within the DNA sequence. I guess I should change that and say DNA sequence. There you go. So that's more accurate. So we're supposed to be cutting the middle of a DNA sequence. So if we have a DNA sequence, for instance, like this pencil right here, we're going to be cutting this section out in between. So cutting that section out in between. Whereas an exonuclease is going to cut off something that's towards the end. So that's the main different groups of restriction enzymes. We also have these methylases that basically can put methyl groups on host cells, so basically prevent the DNA from being cut. So you have certain methylases, for instance, like DAM methylase, that actually will go in, and this DAM methylase will put on a methyl group on the DNA strand and basically kind of protect the DNA from getting cut open and sliced open by these restriction enzymes. So that's pretty cool. Now, when restriction enzymes recognize sites, they're generally recognizing these palindromic sequences, meaning these sequences that read forward and backward in the same way. So if we look at this sequence over here, we have CTG, CAG. You can see that the other, so this is, okay, so we have CTG, CAG, and then the opposite complementary strand reads the exact same way, which is CTG, CAG. So this is a palindromic sequence, and therefore, most likely, it's going to get recognized by some type of restriction enzyme. I guess a better way to put it is that usually restriction enzymes are going to be recognizing these palindromic sequences for cutting, but not necessarily. When it comes to the actual cut itself, the restriction enzyme, depending on the type, will make different types of cuts. So some restriction enzymes are going to be making staggered cuts that result in an overhang with sticky ends. So you have ends that have an overhang over here. In addition, you have certain enzymes that are going to be cutting with blunt ends. So you have these sequences over here where you're just basically slicing it down the middle and there aren't any overhangs. So some restriction enzymes are going to be targeting and cutting and making these straight ends, and others are going to be making those staggered ends. So just two main differences. Now, once you have those restriction enzymes that are cut and you make those staggered ends, for instance, you can actually ligate various sticky ends together. So the complementary overhangs will often allow some sort of base pairing. The blunt ends, of course, can just be simply stuck together. So in order to kind of ligate those strands and form those phosphodiester bonds, you need an enzyme to do that. And oftentimes this is done by something such as DNA ligase. So remember, DNA ligase will come in and basically ligate those strands together, form those phosphodiester bonds. And if we're talking from maybe a recombinant DNA perspective, once you have two different types of strands ligated together, the DNA is basically now recombinant. So that's just a brief description of some of the aspects of basically restriction enzymes. So remember, palindromic sequences are often usually recognized by those restriction enzymes, or I guess maybe a better way to put it is the other way around, which is restriction enzymes generally recognize palindromic sequences. In addition, you can form blunt ends or sticky ends, and these ends can be joined together with ligase, for instance, to make potentially recombinant DNA.